What if some of the damage caused by old cells does not come from the cells themselves, but from the messages they keep sending to the rest of the body? As we age, more cells enter a state called cellular senescence. They stop dividing, which can initially be protective because damaged cells are prevented from becoming cancerous. But many senescent cells do not simply become silent. They begin releasing inflammatory molecules into the surrounding tissue. Over time, this chemical broadcast can disturb neighboring cells, promote chronic inflammation, and contribute to declining function. Scientists have spent years trying to eliminate these cells entirely. But two recent Nature aging studies point toward a different strategy. Instead of killing senescent cells, researchers found that drugs already used in cancer treatment could make them considerably quieter. In aged mice, blocking a pathway involving CDK4 and CDK6 reduced inflammatory signaling and improved physical performance. In one study, the treatment even reduced frailty. That creates a surprisingly simple possibility. Perhaps we do not always need to remove aging cells. Sometimes we may only need to stop them from poisoning the neighborhood. To understand why this matters, first consider what cellular senescence actually is. When a cell experiences serious DNA damage, oncogene activation, or other forms of stress, it can permanently stop dividing. This is an important anti-cancer defense. A damaged cell that no longer replicates is less likely to pass dangerous mutations to thousands of descendants. The problem is what happens next. Senescent cells can develop what scientists call the senescence-associated secretory phenotype, or SASP. Instead of quietly retiring, they release inflammatory cytokines, growth factors, enzymes, and other signaling molecules. These signals can help wound healing in the short term, but when senescent cells accumulate chronically, the same response can become harmful. Imagine a damaged factory that has correctly shut down its production line. That is useful. But instead of remaining closed, the factory starts pumping smoke into the surrounding city every day. The factory itself is no longer producing defective products, yet it is now creating a different problem. Researchers have therefore been pursuing two broad strategies. The first is called senolysis. Senolytic treatments attempt to kill senescent cells selectively and remove them from the body. The second is called xenomorphism. Xenomorphic treatments leave the cells alive but change their behavior, ideally suppressing the harmful signals while preserving potentially useful aspects of senescence. The new studies focus on this second idea. One research team led by Adarsh Rajesh and colleagues noticed something strange inside senescent cells. A protein called cyclin D1 was elevated. That was unexpected because cyclin D1 is normally associated with cell cycle progression, yet senescent cells have permanently stopped dividing. Why would a non-dividing cell maintain a protein associated with cell division? That contradiction became the entry point into the mechanism. The researchers found that cyclin D1, working with the enzyme CDK6, was doing something completely different in these cells. Instead of pushing them back into division, the cyclin D1 CDK6 system helped sustain DNA damage and inflammatory signaling. The process appears to work like this. Cyclin D1 and CDK6 promote accumulation of DNA damage in senescent cells. Pieces of damaged chromatin can then escape from the nucleus into the cytoplasm, where DNA normally should not be floating freely. Cells interpret DNA in the wrong place as a danger signal. A sensor called cGAS detects the cytoplasmic DNA and activates another protein called STING. This cGAS STING pathway is part of the innate immune system. It is extremely useful when cells need to detect viral infection or other threats. But in senescent cells, chronic activation can produce persistent inflammatory signaling. So the chain becomes senescence, increased cyclin D1 and CDK6 activity, more DNA damage, DNA fragments appearing in the cytoplasm, activation of cGAS STING, and finally increased inflammatory gene expression. Then the researchers interrupted the chain. They used palbociclib, a CDK4/6 inhibitor already approved for certain cancers, particularly hormone receptor-positive breast cancer. Palbociclib was not originally developed as an anti-aging drug. Its clinical purpose is to slow the division of cancer cells by blocking CDK4 and CDK6. But here the researchers were using the same molecular target for a very different reason. When aged mice received palbociclib, inflammatory gene expression decreased. DNA damage signals in aged liver tissue also fell. Most importantly, the effect was not limited to molecular markers. The treated aged mice showed better physical performance and reduced frailty. This is where the result becomes much more interesting. A treatment can make dozens of biomarkers look better without producing any noticeable improvement in the organism. Aging research is full of molecular changes whose practical meaning remains uncertain. But frailty is different. Frailty reflects a decline in the body's ability to withstand stress and maintain function. In mice, researchers can assess features related to physical condition, movement, strength, and general health. Improving those outcomes suggests the intervention changed something functionally important, not merely a laboratory number. At almost the same time, another research group reached a remarkably similar conclusion through a different molecular route. Boshi Wang and colleagues studied another CDK4/inhibitor called abemaciclib, also used clinically in cancer treatment. They found that short-term CDK4/inhibition suppressed inflammatory signals in already established senescent cells. That detail is important. CDK4/6 inhibitors are known for stopping cells from dividing, so one obvious explanation could have been that the drug simply prevented more cells from becoming problematic. But these experiments showed that the drugs could alter the behavior of cells that were already senescent. In other words, the treatment was acting as a xenomorphic. It was changing what the old cells were saying. The second team traced the effect through another pathway involving retinoic acid signaling and NF-kappa B, one of the major controllers of inflammatory gene expression. CDK4 and CDK6 interacted with parts of this inflammatory machinery. When the researchers blocked CDK4/6 with abemaciclib, the inflammatory secretory program weakened. They also tested a different molecule that interfered with the retinoic acid receptor RIR-alpha. That intervention similarly reduced inflammatory signaling. Then they moved into animals. In aged mice, both CDK4/6 inhibition and disruption of the related retinoic acid pathway reduced systemic inflammatory signals and improved physical performance. Abemaciclib also improved function in mice after chemotherapy, a treatment known to generate senescent cells and contribute to fatigue and physical decline. Two independent studies were therefore pointing in the same general direction. Senescent cells appear to use machinery involving cyclin D, CDK4, and CDK6 to maintain part of their inflammatory state. Blocking that machinery can reduce inflammatory signaling without necessarily killing the cells. That could matter because completely removing senescent cells may not always be desirable. Senescence is not purely harmful. It participates in wound healing, tissue repair, embryonic development, and cancer suppression. A therapy that indiscriminately destroys every senescent cell could therefore interfere with useful biological processes. A xenomorphic approach could theoretically be more selective in a different way. Instead of asking, which old cells should we kill, researchers could ask, which harmful behaviors should we turn off? That is a much subtler therapeutic idea. Think again about the damaged factory. Senolysis is demolition. Xenomorphism is installing filters on the smokestacks. Sometimes demolition may be appropriate. Sometimes reducing the pollution may be enough. There is another reason these studies attract attention. Palbociclib and abemaciclib already exist. Developing a completely new drug can take many years because researchers must determine how the molecule behaves in the body, identify toxicities, establish dosing, and perform extensive clinical testing. Repurposing an existing medication can sometimes move faster because much of its pharmacology is already known. But this is exactly where caution becomes essential. An approved cancer drug is not automatically a safe anti-aging drug. CDK4/6 inhibitors have real side effects. Depending on the drug, these can include low white blood cell counts, anemia, fatigue, diarrhea, liver abnormalities, infections, and other complications. Cancer patients take them because the expected benefit of controlling a potentially deadly disease can outweigh those risks. That risk-benefit equation is completely different for a healthy 60-year-old who wants to reduce age-related inflammation. There is currently no evidence from these studies that healthy people should take palbociclib or abemaciclib to slow aging. The experiments showing improved frailty and physical function were performed in mice. We do not know whether a safe dose exists that would suppress harmful senescent cell inflammation in humans without causing unacceptable effects elsewhere. We also do not know whether long-term suppression of CDK4/6 pathways could create new problems. These proteins participate in normal cell biology, tissue maintenance, and immune function. Manipulating them chronically is very different from using them temporarily in a controlled experiment. But the studies may still be valuable even if these exact cancer drugs never become anti-aging medicines. The bigger discovery is the target. Researchers now have evidence that cyclin D CDK4/6 signaling helps control the inflammatory personality of senescent cells. That means future drugs could potentially be designed to influence this specific function without reproducing every effect of current cancer treatments. And this connects with one of the largest ideas in modern aging research, inflammaging. As people grow older, the body often develops a persistent background level of inflammation. It is much weaker than the inflammation caused by an acute infection, but it can continue for years. This chronic inflammatory environment is associated with cardiovascular disease, metabolic dysfunction, frailty, neurodegeneration, and many other conditions of aging. Senescent cells are probably not the only source of inflammaging. Aging immune cells, damaged tissues, changes in the gut, mitochondrial dysfunction, and other processes contribute as well. But senescent cells may act as one amplifier. A few damaged cells can influence many healthy neighbors because the signals they release travel beyond the cells themselves. That means suppressing their inflammatory output could potentially have effects larger than the number of treated cells would suggest. The mouse results are consistent with that possibility. Researchers changed a signaling program inside senescent cells, then observed improvements at the level of whole animal physical function. Still, we should not confuse an exciting mechanism with a proven longevity treatment. Neither study demonstrated that CDK4/6 inhibitors extend mouse lifespan. Neither demonstrated that they extend human lifespan. Neither showed that taking these drugs prevents age-related disease in healthy people. What they demonstrated is narrower but still important. Manipulating CDK4/signaling can suppress harmful inflammatory features of senescent cells. And in aged mice, this was accompanied by better physical function. That creates a very different target for future research. For years, one of the central goals in senescence biology has been to find ways to remove old cells. These studies suggest the better question may sometimes be more specific. What exactly makes an old cell harmful? If the answer is partly the inflammatory messages it sends, then aging medicine may not always need to erase the cell itself. It may only need to change the conversation. And that could turn one of the body's oldest anti-cancer defense mechanisms from a source of chronic inflammation back into something far less destructive,